Hello everyone, I wanted to introduce myself. My name is Richard Converse and I'm going to be your online instructor for Bio 119 Current Topics in Biology Lab. By way of introduction, I began as an adjunct in 2004. I completed both my undergraduate and graduate studies at the University of Cincinnati, and after that I began research on biodegradation in the UC Department of Molecular Genetics. I currently hold an MS in Biological Sciences from the University of Cincinnati, and I've got 25 years of research experience in molecular biology. During that time, I've studied plant genetics, biodegradation, muscle development, corneal disease, heart development, brain development, cancer, and the genetics of herpes simplex virus latency. I've worked in bacterial, mouse, and yeast systems during this period, and my interests include gene regulation, evolution, human development, molecular genetics, biochemistry, and of course, teaching. Since 2004, I've instructed courses at the University of Cincinnati, Xavier University, Aunt Nellie College, the University of Phoenix, Cincinnati State Technical and Community College, and Sinclair Community College, as well as Beckfield. The courses I've instructed include biology, microbiology, anatomy and physiology, molecular biology, ecology, and pathophysiology, as well as general biology for both majors and non-majors. I'm certain it will have a rewarding experience as we journey through the field of biology. Please don't hesitate to contact me with any questions you might have over the next 15 weeks. My preferred mode of contact is email, but I also will provide my cell phone number. That is 513-335-1150. My office hours are from 5 to 8 on Friday evenings. I will be at my computer and I'll have my cell phone on during that time. You can reach me by any of the prescribed communication methods. Now, what I'd like to do to start off is to introduce you to the course you're going to be taking. This is Biology 119, Current Topics in Biology Lab. It accompanies Biology 120, which is the lecture component, also delivered online. In this course, we'll conduct an at-home, hands-on investigation of major biological concepts as they relate to current topics in the news. This course will be entirely online with a home lab kit provided for you at a nominal cost to facilitate your exploration of biology. This course will be broken down into 13 modules, each designed to explore a critical biological concept. You'll be expected to perform the experiments as outlined in the instructions that are provided in your home lab kit and report your results and observations by posting them to the Canvas Learning Management System. There will be 3 online lab practicals that will require that you study the assigned experiments as well as the results pooled from the class and posted on Canvas. These will be timed, they'll be 90 minutes in length, and the questions will be a combination of short essay, true, false, and multiple choice. You will only have a single attempt at each of these practicals, so to avoid any technical issues, I'm going to deploy a practice quiz during the first week of class to be used as both a syllabus review and as a method to work out any bugs you might experience using your computer with the Canvas Learning Management System. Be on the lookout for this practice quiz during week 1. It's highly suggested that you use the Firefox web browser. You can download that at www.firefox.com. There are versions both for the Macintosh and Windows operating systems. Now, let's explore each of the modules you're going to work through during the next 15 weeks. So our first module deals with the scientific method. Basically, how is scientific knowledge obtained? In this module, Research in the Scientific Method, we'll establish and execute the steps of the scientific method utilizing the home lab kit that accompanies this course. You'll devise a hypothesis, design an experiment, gather data, analyze this data, and determine if that data supports or refutes your hypothesis. Again, as you record your data and shape your hypotheses, you want to follow the steps of the scientific method as outlined in the lab materials provided. Our next module is going to deal with biological macromolecules. Essentially, we're asking the question, what is the chemistry of life? In this module, we'll address the role of DNA, proteins, and lipids in the living cell. You'll explore the chemical properties of these macromolecules and elucidate their role in living systems. The home lab kit will provide the necessary materials and experimental protocols, and as with all the modules, you'll be expected to post that lab report to Canvas at the conclusion of those experiments. Our next module deals with the effect of temperature and pH on enzymes. So we're asking here, what is the chemistry of life? In this module, we will address the concept of optimal conditions for enzymatic reactions. We'll investigate the role of temperature and pH on enzymatic processes, as well as address the concept of enzyme substrate specificity. The home lab kit, again, will provide the necessary materials and experimental protocols. You're essentially going to learn how it is that all the reactions that sustain living systems are facilitated. And the tool that accomplishes that is the enzyme. In our next module, mitosis and meiosis, we're going to elucidate how cells reproduce. We'll investigate the phenomenon of cell division in the context of growth and repair, and this will be the mitosis portion of the module, as well as the process of gametogenesis, which is how eggs and sperm are produced. The home lab kit, again, will provide the necessary materials and experimental protocols. In our next module, we're going to examine the role of DNA in traits. Essentially, we're asking, where do traits come from? We'll address the role of DNA in directing and influencing traits in living organisms, and the home lab kit, again, will provide the necessary materials and experimental protocols. In our next module, genetics and genomics, we're going to determine the role of DNA in determining the types of proteins that living cells produce. Essentially, proteins are the workhorse of all biological processes, so the information that goes into constructing proteins is critical in maintaining the living system known as the cell. So we'll describe and demonstrate the role of transcription and translation in living systems. This is a commonly referred to phenomenon as known as the central dogma of biology, and it describes the method of information flow in a living cell. The home lab kit, again, will provide the necessary materials and experimental protocols. In our next module, cell membranes and transport, we're going to ask the question, how do cells obtain what they need in order to live? We'll investigate the various processes employed by the cell to permit substances to move across the cell membrane in order to sustain life processes. And again, the home lab kit will provide the necessary materials and experimental protocols. Once we've addressed the chemistry of life and the mechanisms that govern the smallest unit of living matter, the cell, we're going to move up a level and we're going to examine the human body. And our first physiology module is called homeostasis. The question we're asking here is, how does the body maintain conditions that are compatible with life? We'll examine the concept of homeostasis, which is the body's internal balance which serves to maintain conditions compatible with life. The home lab kit, again, will provide the necessary materials and the experimental protocols. We're going to continue our physiology series with a laboratory on water, electrolyte, and acid-base balance. The question we're asking here is, how does the body maintain proper fluid balance and pH? And why is this important? In this module, we'll examine how pH and water balance are modulated in order to maintain homeostasis and create conditions compatible with life. And again, the home lab kit will provide the necessary materials and experimental protocols. In our third physiology module, chemoreceptors, we're asking the question, how are chemical signals sent and received in living systems? In this module, we'll examine how receptor-ligand specificity contributes to normal cell and tissue function. Essentially, we're going to elucidate how it is that different cells and tissues talk to each other in the context of a living organism, and what molecules are involved in that communication process. The home lab kit, again, will provide the necessary materials and experimental protocols. In our fourth physiology molecule, nutrition and metabolism, we'll be asking the question, how is the energy in food turned into energy for life? We'll examine the components of a typical diet and investigate the calorie content of these components. And again, the home lab kit will provide the necessary materials and experimental protocols. In our fifth physiology module, respiration, we will investigate the chemical process that extracts energy from macromolecules and transfers that energy to fuel immediately available to do work. So the question we're asking here is, how is the energy generated for the work of life accomplished at the cellular level? We understand that we have to eat food to live. We learned that in the nutrition module. Here, we're asking questions about the chemical processes that transfer that energy into a form that can be used to sustain life. Again, the home lab kit will provide the necessary materials and experimental protocols. In our final module, microbes, we're going to ask the question, what is the scope and role of microorganisms that populate our environment, and how can we detect them? We will investigate the scope and variety of microbial life in your environment and describe methods used to measure microbial growth and the composition of microbial populations. The home lab kit will provide, again, the necessary materials and experimental protocols. Okay, now that we've gotten things started, I'd like to ask you to do a few of the following things to prepare for the course. First, download and read the course syllabus. Pay particular attention to the course policies and take note of the information provided on the various ways that we can communicate with one another during the next 15 weeks. Second, make sure to get the home lab kit, including the manual. You can order this kit using the link available on Canvas in the introductory module. We're going to be using the kit right away, so make sure that you have it in time for the start of class. The class moves very quickly, so please don't delay in purchasing this material. Third, go to Canvas and start the first module. You'll see instructions when you get there. Your first assignment is to go to the discussion section for our class on Canvas and post a short introduction. You'll find the instructions on the Canvas assignment link. I want to thank you again for joining me for this course. I'm looking forward to getting to know each and every one of you. If you have any questions, don't hesitate to send me an email or contact me by cell phone. Again, that number is 513-335-1150. Thank you very much.